Hi everyone, so today we're going to be looking at A2.3 viruses in IB biology, and this is only for the higher levels. So let's start by understanding common structures of viruses. Viruses are very diverse, but there are some structures that are shared between all viruses. So if you don't know yet, viruses infect cells and they reproduce inside them. The first common structure is that they have a small size. Again, as I said, they invade cells, so they need to be smaller than them to be able to enter them. They also have a fixed size, so they don't grow or increase in size. They just assemble inside a host cell and create more viruses, but the viruses don't increase in size themselves. They use nucleic acids as the genetic material. Why? They are using the cell to replicate their genetic code, so they need to follow the same rules as a normal cell would. That's why they use the universal genetic code. Remember the codons, the three bases. You can remind yourself of that by checking out the nucleic acids video. But yes, again, having nucleic acids as genetic material is essential since they use the host to synthesize proteins and replicate their material. Then they have a protein capsid, and this encloses the genetic material in the virus, and all viruses have this. And then they don't have a cytoplasm, and they don't have enzymes, or if they do, they have very, very few. They use the host's enzymes, and if they do have some, those are essential for replication or for infection, but that's rare. Viruses are very, very diverse. It's thought that they evolved at many different points. We'll look at that in the next slides. But this means there's a lot of diversity. You don't need to understand either of these images in detail, but just know that the main diversity comes from in genetic material, so they can be DNA or RNA viruses. Then they can also be single-stranded or double-stranded. They can have different lengths of their DNA or RNA. They can be circular or linear, and this all contributes to differences in how they replicate inside the host cell. So very varied. The processes are very different. And then another big difference is they can either have an envelope or not have an envelope. So this is when the virus is leaving the cell, after it's already invaded it, let's say, the virus can use the host plasma membrane to create an envelope for itself. And then this can help it invade other cells. But this is not always the case. Some viruses are enveloped, some are not. All right, just remember the two main reasons for diversity. Now, a virus normally has two different life cycles. So, for example, the bacteriophage lambda infects E. coli. This is what's represented in the image, right? And once it enters, it can follow the Lytic cycle or the lysogenic cycle. The gist of this is that the Lytic cycle is where the virus reproduces and bursts out of the host cell, so it kills the cell. In the lysogenic cycle, the DNA of the virus is integrated into the DNA of the host, in this case the bacterium, but it doesn't produce new viruses, so it just remains there, right? The DNA is integrated and remains there. Why would it do the second one if it doesn't kill the host cell? You'd think it would want to kill the host cell. The problem is, if they kill the whole host, then they can't reproduce. So viruses aim to invade cells without damaging the host too much. Also, if you're not bursting cells, you might avoid the immune system. So it's good to avoid recognition. Let's start by looking at the Lytic cycle. So this is how a normal Lytic cycle might look like. In the case of the lambda bacteriophage, this is the example we're looking at. So the virus attaches to maltoporin, which is on the outer membrane of E. coli. Again, E. coli is the bacterium. Then the DNA of the virus can enter via maltoporin because maltoporin has a pore in it, as the name says, right? It's normally used to absorb carbohydrates, but the virus takes advantage of this. It's not like the cell has a space for the virus to enter, right? The virus is just really smart and knows how to use a normal structure in the cell. Then the viral DNA ends will join to form a circle. This is then replicated about 100 times. This is how we create new viruses. Then this new DNA is transcribed to form mRNA. You don't need to understand this in great detail, but then the mRNA is used to synthesize proteins using the host cell ribosomes. And then some of these proteins that have been assembled, this is all linear, right? This all follows, the steps follow each other. These proteins will then self-assemble to form capsids with the viral DNA inside each capsid. Again, then the viral proteins can make holes in the membrane of the host cell, causing cell lysis, so the cell bursts, and the viruses can then spread to other cells. The lysogenic, on the other hand, doesn't cause death to the host, and it causes very minimal harm, so it's almost undetectable. In this case, again, very similar. We attach to maltoporin in the same way as in the Lytic cycle. The DNA enters, but now instead of replicating the DNA, the viral ends are joined, so again, it forms a circular DNA, and then it's integrated into the host DNA. This is done through the enzyme integrase. And now, this is an important term, the virus now exists inside the host DNA, and it's now called a prophage. And now, every time the bacterial host replicates its own DNA, it will also replicate the prophage with it, aka the DNA of the virus. It's important to note that at some points, you can go from lysogenic to lytic. So you've integrated your DNA, and then the virus changes configurations, and you can start creating lysis for an even more harmful effect. Good. Now, what about the evolution of viruses? So we can kind of assume that if viruses need cells to replicate, because they are parasites, right? They need them. Cells must have evolved before viruses. Also, viruses use the same genetic code as cells. So this adds even more evidence to the fact that viruses emerged from cells. Another thing to note is that because viruses are very diverse in their structure and genetic constitution, viruses probably originated at different points rather than having a single common ancestor. So what are the two main hypotheses for how they evolved? Well, there's two, the progressive and the regressive. The progressive hypotheses say that viruses would have come from cells through a series of modifications, right? So progression, modifications. This fits with the fact that there's retrotransposons in cells. So in our cells, there's these are parts of DNA that can, it's quite complicated, you don't need to understand it, but it's parts of DNA that can replicate and then jump between our DNA. And then the regressive hypothesis is similar, but instead of modifying cell components, it's the theory that cell components were just lost over time. And this fits with the variation in complexity in viruses. If a cell would have lost less things, it would have led to a more complex virus. If it lost more things, it would have led to a simpler virus. For example, poliovirus is small with very few components. But then other viruses, such as mimivirus, are much larger and complex. So those would have lost less items in the cell. Great. Let's stop here for a question, a paper one question. So what distinguishes the lytic and lysogenic cycles of viruses? This should be really easy. Again, pause now. But the answer is B. The lytic cycle always causes host cell lysis. The lysogenic does not. D, which says the opposite, is just not true. And then A and C are incorrect because in both, the virus enters the host cell, or rather the DNA of the virus. But regardless, so it's B. Great. Okay, so apart from where they might have evolved from, it's very important to understand that viruses have very rapid rates of evolution, so they change very, very quickly. Why? Three main things. They have quick generation times, so you can create new viruses in less than an hour because they replicate super quickly, which means you get changes in the virus all the time. Then they also have really high mutation rates, and that's how you create diversity in the genome. And they have very high natural selection. Why? Because we have an immune system that's out to kill them, right? So this encourages rapid evolution. Let's look at two examples. So we have influenza and HIV. HIV has very high mutation rates because it has a reverse transcriptase enzyme, which does not proofread. So it makes a lot of mistakes, basically. These mistakes are mutations, which then lead to drastic changes in the virus. This is why HIV is a chronic infection and there's still no cure, because it changes so quickly that by the time the immune system gets used to it, it's already changed. And then influenza is kind of similar. Influenza has an RNA replicase, that's the enzyme it uses, which also does not proofread. So it also has a very high mutation rate. And influenza also has 8 separate RNA molecules. So instead of normal viruses have one genetic material molecule, one DNA molecule or one RNA molecule, but influenza has 8. So it can combine them to create very different combinations, and again, evade the immune system. Also, influenza can be transmitted between birds and humans, which means that new strains occur all the time. And influenza, if you haven't realized yet, is what causes flu, right? So the flu vaccine needs to be updated every year because it changes so much. Okay, great. So that's all for the content. This is a really easy topic, so that's good. But let's do one last question before we go. Again, easy. What are common structural features of viruses? And pause now, but the answer is C. 1 and 3. So all viruses have the protein capsid and they all have no cytoplasm. However, not all of them have RNA. Some of them use DNA instead. Okay, good. I feel like I breezed past all of this. So if you do have any questions, please let me know in the comments. But I'll see you next week for the next topic. And in the meantime, have a good week.